standards for building very complicated machines in bacteria. So if you go back historically, it was a real boon to be able to say, I'm going to create a plasmid that will make E. coli make this, and I'm going to see what happens. But the current approaches become rapidly very complicated when it's not just, I want to add this one thing. I want to add many things at the same time and see how they all work together. And to really make complex and interesting machinery, and machinery that is reactive, like if you look into some of the simpler combinations of parts, they'll talk about how this functions as an AND gate, or how I can control the activation of this if both of these are present but not either one. To do that, I need to be able to sort of install into the E. coli chassis a bunch of different parts at the same time. And that's not easy. And it's made more difficult when everyone is kind of doing those things in different ways. So one thing IGEM is doing is it's kind of trying to find the best way to do that in a very standardized way, so that if you develop a part, it can be used by anybody. Because everybody else who understands that standard can go, oh, you have a part that does that? I can use that in my machine now. And also they're just using existing parts and new parts that they're developing to make increasingly complicated combinations and machines in E. coli. So they're trying to take advantage of these new techniques and these standards to do things that would be extremely difficult to do outside of having the BioBricks type way of thinking. If you didn't have something like that, it would be almost impossible to do. There are thousands of ways to do it, and most of them would take a lot longer than BioBricks.
So, what is this year's UC Berkeley IGEM project?
So this year, UC Berkeley is working on sound-initiated lysis, where we're going to track down a promoter that is sound sensitive. And we know from research in the field that there are ultrasound sensitive promoters, and there's some other evidence that even audible frequency sound can initiate transcription of certain genes. So if we can figure out what changes in the cell initiate that sort of gene transcription, then we can build a part that will be turned on at the same time. And that part is going to involve a phage system for lysis. And part of that project is going to be basically setting up, you play music or perhaps expose cells to ultrasound, and the cells will lyse themselves. They'll basically destroy themselves. And the advantages there is, at some point, if you have another part in that bacteria that is making something that you want, you have to break the cells open to get at it. So if there's a product protein that you're producing and you want to purify it from the E. coli, you don't have to go through a long step where you break apart the E. coli. And some of those treatments are very harsh, so you may actually damage your protein getting at it. We're hoping that this technique will not only be really easy to initiate, you just expose them to sound, but it will also be less harsh. And because of those advantages, we're looking into some specific applications, including the gateway reaction, which is a commonly used reaction for switching out parts between different kinds of plasmids.
And maybe you want to, I don't know, explain the gateway reaction a little more, if that's possible.
To be honest, without a board, trying to explain it in words is just going to be painful.
Okay.
Well, if you want it to draw it, I can film it.
I would actually have to go grab a paper from downstairs. Like
okay.
the drawing of what gets switched out is pretty complicated. There's a lot of different parts of it, and I couldn't draw it. I could draw it from memory, but I would likely make a mistake.
Okay.
You can probably just have like a flashing, like, click here to see what gateway, the gateway thing is all about.
Yeah, the group actually is working on a flash animation for how that works.
Okay, yeah. That's
So
fine.
you could link to that. That'd be really good.
Mm-hmm. Or just edit it into the video. Or,
That would be fine. Talk. Oh, you'll be around. Yeah. I don't know who's gonna. I know who started, and I don't know who's gonna finish it.
It's kind of exciting. Flash animation.
They're also working on one for the logo for the team, which is, I don't know if have you seen Molly's?
With the thing sitting in the chair.
Yep.
Yeah. Yeah, it's a good idea. Okay. Okay, so one question I had was, do you have any ideas of what difficulties you anticipate with this project?
Well, this project, difficulties associated with the project. I expect at least one important thing will not work. And which is why, just like with any scientific project, you don't pick a single direction and say that's all I'm really looking into. And we imagine what the perfect system would look like, but we've also, when we're thinking about it, we imagine several imperfect systems. Like if this goes wrong, then we can still do this, and that will be useful. And if this goes wrong, well, we can still do this, and this will still be useful. We're looking at 2 or 3 different directions for this system, for this way of lysing cells and taking advantage of hopefully the less harsh conditions and the fact that you can just tune it on by hopefully turning on a stereo. And of those directions, one will probably fail, maybe even 2. It's an engineering problem, and you very often you have a lot of different choices in how you can put these things together. And there are parts of the system that you're very comfortable with, and other parts of the system where, to be honest, you just have to try it and see. And we are right now approaching the try it and see stage. So what's going to happen is that we put together our wish list of parts, and the IGEMers have been developing those parts. And what we're going to do is test them individually or in small groups, and identify where the problems will be. And then we'll probably have a meeting in the next couple of weeks where we'll sit down and go, we're comfortable with these working, these might be an issue. Can we either make these not an issue by performing different experiments or hooking them together in a different way, just doing something different? Or if they are going to be a consistent problem, how do we take advantage of the stuff that does work?
And these are basic parts, which means they don't they don't yet have a function. They must be strung together in order to have that functionality.
Yes, the basic parts. For example, if you wanted